So despite the fact that you guys are just learning about the concept of meiosis, I'm going to take us a step further and look at a problem associated with meiosis not working properly. Now, the good thing is that by talking about this, it is going to allow you to do better on the group quiz because I'm revealing some things. But know that it might be a tad difficult for you to follow exactly what's going on. We'll see. Let's start off with a cell. And in this cell, we're going to have only 3 pairs of chromosomes. I want you to notice that in this cell, I haven't drawn a nucleus because frankly, it makes this a lot easier. And also, I want you to note that the homologs are not right next to each other. For example, if you look at this one, its homolog is way over here. This one, the homolog is sort of close, but it's not snuggled up next to it. And in this situation as well. So homologs don't need to hang out next to each other. They just need to be the same length. That's really the key element to this thing. And one of them needs to be from mom and one of them needs to be from dad. Now, in the process of meiosis, the homologs are going to be duplicated. So let me take a moment to duplicate each one of these. So if that's the case, then this is happening after DNA replication has taken place in the S phase of the cell cycle. The next thing that happens is that each of these homologs will actually pair up with each other across from each other in the equator or along the equator of the cell, like so. Now I could draw the equator, but I don't want to because it makes a mess of things. But understand that the equator basically runs right down the middle of the cell. And if that's the equator, then the points that I'm going to put little red stars on, or asterisks, those are the poles. And the poles are from where the spindle fibers will run out to attach to the kinetochores on these chromosomes. Okay, so we have spindle fiber attaching here, spindle fiber attaching here, spindle fiber here, same thing here, just like that. All right? And this is a very important interaction because if we were to blow this up, we would know that the spindle fiber attaches to something called the kinetochore, which is hanging out right there at what's called the centromere, the waist of these chromosomes. And in fact, there are 2 such kinetochores that are connecting right here and right here. And these spindle fibers, there's not just single fibers like I'm showing it here. There are multiple fibers, and they'd be attached to both of the kinetochores. Now, what's important for you to understand here is that the spindle fibers here are all going to shorten, and they're also, strangely enough, there are spindle fibers in between these things that will extend and get larger. And ultimately what that means is that this blue thing right here, this blue chromosome, is going to be pulled towards this pole. This chromosome will be pulled to this pole, etc., etc. Okay? So in fact, if I were to show this in the next, I'm sort of going to do like a Julia Child cooking thing. I'm going to show you what happens after the spindle fibers have done their job. It's going to look something like this. There you have it. And then eventually what's going to happen is that let me just erase some things in here. You're going to have the cleavage furrow, if this is an animal cell, that will develop here. And then eventually it's going to seal it entirely so that you'll be able to break these things into 2 separate cells. And let me show that with the next thing. So you can see here that I've just created 2 separate cells in here, but I'm not done because in meiosis you have another one of these alignment events and the chromosomes will separate themselves across to the left and right pole in this case, similar to what I'm going to show you right now. Before I head over there, I want you to note here that if I'm going to split these things up further and still have the same amount of information into both cells, then I'm going to have to break the sister chromatids, snap them at their centromeres to drag them to different sides. So in fact, if we take a look at this, that means that instead of having this duplicated chromosome, that goes away, and when I snap it apart, I'm going to have one chromosome on one side, one chromosome on the other. They were once sister chromatids. Similarly, when I set this all up and break them apart, instead of having this and this, I'm going to have a blue medium-sized one here, another blue medium-sized one here, and a tiny blue one here, tiny blue one here. And then, as you can imagine, we're going to have the process of cytokinesis, which would take place, right? And it would split these things ultimately into two separate cells. Same thing would be going on over here. I'll have two long blue ones on either side, two medium pink ones on either side, and two small pink ones on either side. So that ultimately, it's going to look something like this. I have 4 of these cells, 2 of which look very similar to one another, right? So this and this look like they have the same genetic component, and this one look like they have the same genetic component as well. In fact, you'll find out that they don't, that even this one and this one are different, but I'm not going to go into the details. What you should know, though, is that ultimately I'm going to have 3 chromosomes in each one of these resulting cells. And these are all going to be sex cells. If it's a female, these will be egg cell and 3 polar bodies. If this is a male, we're going to have 3 different sperm. So I have 3 chromosomes. I'll just write 3CH in each one of these things. 3CH. They're not all the same. It's not the same combination, but certainly you need to have a little one, a medium-sized one, and a long one. So here's the thing. The separation of the chromosomes is enormously dependent on the spindle fibers connecting correctly. What would happen if the spindle fibers didn't connect to the kinetochores properly? I want you to imagine this. So we have this beginning situation that we have right here, but instead, for some reason, there fails to be a good connection between those two. The pink medium-sized duplicated chromosome and the pole. And in fact, instead of that happening, a spindle fiber comes out and attaches from the wrong side, the left side pole, to that pink one. I think you can see that it's going to quickly be a pretty significant disaster, because we're going to bring those 2 medium-sized chromosomes over to one side and have no medium-sized chromosomes over on the right side. So it looks something like this. Do you see how already at this first split we're going to have too many chromosomes on one side and not enough chromosomes on the other? And if we carry this to the next stage, you can see here how we're going to, if we look at this situation, I've already split up the chromosomes. Here I haven't yet split it up, but let's go ahead and split that one too. So you can see I have these just about done with cytokinesis, not enough chromosomes. And I think you can predict what this is going to look like with the final gametes, the sperm and the egg. You can see in this situation, it's a bad situation. If we want to assign numbers to each of these chromosomes, we can see that we have a chromosome 1, a chromosome 2, another chromosome 2, and then a chromosome 3. It's the same situation we have going on here, right? 2, 2, 3, and 1. And then in this situation, we do have a chromosome 3 and a chromosome 1, but we're totally missing chromosome number 2 in both these cases. That is not a good thing. Now, keep in mind, you have 46 such chromosomes that need to be sorted properly in the very beginning with the germ cell. So there's a lot of chance for spindle fibers to attach in an improper way. And in fact, there's an even greater chance for spindle fibers to fail as the woman grows older. So this sounds funny. Why am I saying just the woman? Doesn't this happen in the formation of sperm? Well, actually not so much, because really we care so much more about the women's eggs. Don't forget what I said today in class, that the eggs are in a woman from the moment she is born. They aren't fully developed, but they're there, which means that they can age. And when cells age, then the proteins and all the other stuff that's going on inside of the cell also ages. So in a man, you're constantly making new sperm, and so it doesn't age in the same way. You're not going to have bad spindle fibers that have been sitting around for 40 years being attacked by chemicals and a bad lifestyle and radiation, etc. So when this happens, when this process happens, we refer to it as being a non-disjunction. Okay, it hasn't properly broken apart, non-disjunction. And non-disjunctions lead to something called aneuploidy, which is what I have up here. So the state of aneuploidy is when you have an improper number of chromosomes as a result of a non-disjunction event. Now keep in mind, I've shown here the non-disjunction event happening right here, but the non-disjunction event could actually happen at this stage too. Let's look at this as if it had a proper initial disjunction going on here, but then when they align in the next phase, like I'm showing here on the left, imagine if we have the issue happening now. So here we have proper attachment, but here we don't have attachment to the chromatid associated with this duplicated chromosome. So when they pull apart, it's going to look something a little bit different for that one. Let's imagine that we have everything over on this side right here looking proper. So that means I have chromosome number 1, 2, and 3 setting up just fine because they had proper disjunction going on here. But over here, I have a non-disjunction event. So what is that going to look like in the final cells? You might have been able to predict what we have going on here. Because in this case, instead of having the 3 chromosomes I'm supposed to have, I have 5 chromosomes over here, right? Yet I have, and I have 3 chromosomes in this one here, and this one here. So in this case, it's a little bit less catastrophic because the non-disjunction event happened at this stage of meiosis as opposed to happening up here at this stage of the meiosis. That may be a little confusing and hard to understand, but hopefully as we go through this process, you'll understand it a little better. I've run out of time, so I'm not going to talk about karyotyping in this situation, in this screencast. I'll leave it at this concept of the non-disjunction and the aneuploidy. Okay, hope you get this.